In this module, we will discuss the advantages and the use of another branch of chromatography, that is the adsorption chromatography. Now, what is adsorption chromatography? The basic principle of this adsorption chromatography is that certain materials which are collectively known as adsorbents. There are some materials which are known as adsorbents. They adsorb, have the ability to hold molecules at their surface. That is, the molecules, certain molecules, they can be held by these adsorbents and on the surface. Now, how they can hold those compounds on the surface is that they involve weak, non-ionic, attractive forces of the van der Waals type and hydrogen bonding type that occur at specific adsorption sites. So there are the principle or adsorption process for these adsorbents that involves weak and non-ionic attractive forces of the van der Waals types. You know, between the molecules, there are certain forces which act upon those molecules. They are sometimes weak and are known as van der Waals forces. For example, between hydrogen bonding type. In water molecule, you see the hydrogen bonding occurs, and these are the weak bonds, and that occur at specific adsorption site. You see, on that adsorbent, there are sites where they can hold those molecules for which they are the adsorbent by the van der Waals forces. Now, the adsorption sites, they have the ability to discriminate between types of molecules and are occupied by molecules of either the eluent or of the analyte. So you see, it is not this that these adsorbents, they will just adsorb every molecule. No, they are very specific, and they have the ability to discriminate between types of the molecules which they want to adsorb and are occupied by molecules of either the eluent are those molecules which just wash them away or which remove them from those sites, or the analyte, that is, the analyte are the molecule which are adsorbed on those molecules of adsorption. An eluent is passed down the column. Differences in these binding strengths, they lead to the separation of analyte. So what happened? That when on the adsorbent, different molecules, they are absorbed in the column, now you have to remove those adsorbent. So you pass down the different compound or different strength of those molecules or eluent which removes them from the binding sites. For example, the different chemicals or different material rather which can be used there. The silica is one of that, and it is the typical adsorbent and has the silanol, SiOH, silanol, silicon OH molecule group on the surface. That is the silica base. Now, silanol, they are slightly acidic, as we can see from the composition of the molecule, and they can interact with polar functional groups of the analyte or eluent. That is, they are slightly acidic they can interact with polar functional groups of the analyte or the eluent. And then the topology of these silanol groups in different commercial preparations of silica explain the different separation properties. So you see, commercially, these adsorbents, they are available, but how that there are different group differences which they are commercially made, and they can be used to adsorb commercially different adsorbents so that they can be used. Other commonly used adsorptions are alumina, aluminum hydroxide, and carbon as such. Now, in general, an eluent with polarity is comparable to the most polar analyte in the mixture is chosen. That is, that eluent with a polarity, if the eluent is a polar compound, then it is comparable to the most polar analyte. Analyte kya? Which is adsorbed on those molecules or in the mixture. Now, the mixtures of solvents, they are commonly used in the context of gradient column. So you see, in that adsorption chromatography, where we use it as a matter of fact to remove or to separate the adsorbent which can be adsorbed on certain adsorbent molecules, and then through eluent, when we pass that eluent through that column, they are just separated. What happened? Because eluent takes the place of that pore where the adsorbent material has caught or has adsorbed the analyte. So it releases that. This is a technology which is frequently used for the preparation of and the purification of chemicals, of materials which are of commercial interest or which are of research interest.